Stallings, talk about fish and ecology. And we thank him for coming back from Tampa this morning to give this talk.
Yeah, I make the drive over to Tampa every Friday to teach an undergrad class, which really makes me value how nice it is to come here. But thank you guys for coming back from lunch. I hope to share a little bit about what we're doing in the Fish Ecology Lab with you guys today. We have grown to be a fairly large group of people, and I wanted to highlight a little bit of what everybody's doing in the lab. So my goal here was breadth instead of depth, so you get a brochure overview of the Fish Ecology Lab. As I gloss over some of these studies, if anything grabs your attention you want to hear more about, please come talk to me. Better yet, talk to one of the students or postdocs that is doing that work. You can also go to our webpage. I don't know that you're going to get more information from the webpage right now. We need to update it. We just talked about that at lab meeting on Wednesday. But you can get there. There's the URL. There are multiple links from the CMS webpage. You can check that out. We are also on social media. We have a Facebook page, which we started about a year ago. We have over 350 people that like us. We like to bump that up a little bit, but that's pretty good. And each week we post some original material. We sometimes repost other things, that we think are cool from the ocean environment. But most of the stuff on Facebook is our own original material that talks about some of our research, some of the things that we've accomplished, etc. Over the winter break, I signed us up for Twitter. And obviously here, we've tweeted 8 times, so I'm still trying to work my way through that. I'm finding that I actually really like Twitter. I was just checking it. Where's my phone? I actually really like Twitter. I've never done it before, but it's like an injection of news that you get, really fast and really up to date. So you can follow us on each of these. We somehow got Fish Ecology Lab for both instead of Fish Ecology Lab _8398, whatever. I guess that indicates that fish ecologists are sort of behind on social media in general. A little history about the Fish Ecology Lab. I came here in fall of 2011. That was my first semester. 2 students started with me that first semester. And then we slowly grew, added a student here, a student there, then underwent very fast, rapid growth, and then seemed to reach carrying capacity here. And we've leveled off. I certainly think we're at carrying capacity. I don't know that we can add more. And right now we have 3 postdocs. 2 people have completed their master's degrees. One undergrad has completed an honors degree. She's not actually included with these data. And then 7 current students. And here's the good-looking group here. I had to paste in a couple of people. We took this photo a couple years ago. It's almost like, what am I looking at? So a couple of our newest members here, Butch and Tim, had to be pasted in. Everybody's currently a member except for Brittany. She just finished in the summer. She's not in here by any chance, is she? No, she knows what we're doing. She's over at FWRI. And then Denora also finished last summer, but she decided please sir, can I have some more? So she stuck around for her PhD, which she's just starting now. And I'm really proud that Denora stayed around because I think that says something about what we're doing here in the college, at the university, and hopefully it says something about what we're doing in our lab, because she did have another really good opportunity, and she chose us, so I'm proud of that. Okay, notice one thing, I say fish ecology with parentheses. I'll show you why later on. I know that part of the impetus of this is to inform especially new graduate students about the research that we're doing at the College of Marine Science. When I was first starting grad school, this paper came out in Science by Jeremy Jackson and colleagues, and in it he described how history had repeated itself over and over again in coastal ecosystems around the world, where humans would come in and colonize an area, their populations would grow, and as they did, a series of disturbances would occur, often in the same sequence, starting with fishing and often over exploitation of resources, followed by pollution, loss of habitat, and then later on down the road, introductions of non-native species and more recently climate change. And all of these individually can lead to altered ecosystems, and in an additive or in a synergistic effect can have a really strong influence. And a lot of what we do in our lab can in some way be tracked back against this backbone that I'm going to use for the remainder of the talk. So most of my work has focused on the fishing part of this backbone. And so I'll work through some of the case studies of work that we're doing in the lab. First of all, when I was a graduate student, I wanted to know what the status of predatory reef fish populations was across the greater Caribbean. And this is really difficult information to gain from areas that don't have really strong centralized fisheries management agencies like we do here in the United States. So I had to use a variety of different approaches. I'll spare you the details for time. But the end result is that the more people in an area across that greater region, the fewer predators that we saw. And this applies both in total abundance and in richness of predators. We also saw that the structure of their communities changed. And that was related to the number of people there. And this is most likely linked back to overfishing those big predators. Fish out those big predators first, and once those are gone, you shift down to slightly smaller predators, etc. And so combined, what this meant was that in areas with a lot of people, we see reefs with very few predators, only a few species, and those species tend to be the smallest ones. Whereas areas where we have fewer people, we have less disturbances, we have intact predatory communities. Okay? Now, on that brochure I had on the title page, you might have noticed it said Integrating Basic and Applied Ecology. And part of what we're trying to do there is we go, okay, well, what does this mean to the system as a whole? What does it mean to have all of our predators versus a few of them? And so we went in and did some experimental work and found that having those large predators around is really important. They can have effects on the behaviors of the other predators, which might have, themselves might have negative effects on the lower trophic level prey. And so this lends itself to some management discussions that we can have about whether we're going to manage for just this component of the human fish socio-eco realm of this model. Are we going to manage for this entire thing? Because through our experimental work, we've shown that humans can have some indirect, negative, and really some unanticipated effects on marine ecosystems. Okay, so I continued to do some work on large predators, a little bit closer to home. And this is some work that we're continuing to do on Goliath Grouper, which is one of the largest reef fishes in the world. History played out again with Goliath Grouper. They were overfished, which reduced their size structure and their abundance, all the way to the point that their population crashed. And the US federal government enacted a fishing moratorium on them in 1990. And we're not all doom and gloom. There's some signs of recovery here. There's some signs of hope in US waters. Now, they're still critically endangered throughout their range, but there's some positive signs of recovery here in the United States. And they're still protected, by the way. However, not everybody is a big fan of this. How many people in here are avid recreational fishermen? Anybody? I know you are, Ben. If you're an avid recreational fisherman, you've at least had conversations with some of your friends. I've had conversations with this guy about it as well. He's wrong. And that's the Goliath grouper are gobbling up all the other fishes that we can catch and keep on these reefs. So smaller groupers, the smaller snappers. Okay, so it doesn't take you long to type in Goliath grouper recovery Florida in Google and get these kinds of headlines. Some of them are fairly hyperbolic, and some people would even say they're going to crawl out of the ocean and eat our babies. So one of the studies that we currently have going on is to better understand the status of the US Goliath grouper population. And because we do think that they're still in a recovery phase, we wanted to understand their status in a non-lethal manner. So most of you guys that have taken biological oceanography or taken some kind of fisheries course know that one way to determine the age of a fish is to remove their otoliths, the ear bones in their heads, and from that you can age them just like rings on a tree. If you're digging in the head of an animal, you're probably going to have to kill them, right? So if they are just in their early stages of recovery, pulling out their otoliths is going to fly in the face of conservation desires. So we developed we didn't actually develop it, but we have demonstrated that using dorsal fin rays works just as well as the otoliths for the Goliath grouper. And so we're able to get this information about the age structure of the population, which is really important. If you only have a bunch of young fish in a population, that's not a very healthy one. So this will help us get a better grip on the age structure. We're tagging them. We've got various types of tags out there to look at movement. We're looking at their spawning potential. Anytime you're thinking about the status of a population, trying to predict what they can provide to the next generation is really important. And then we're also trying to get at this whole issue of, are they really gobbling up all those fish that we can catch and eat? And we're doing that. You don't get a full appreciation here, but this guy has his hand in the mouth of this fish. And you can imagine, this looks like a smaller Goliath grouper. You can imagine with the one that's a little bit longer than that, how far in you have to get your arm to get down to the stomach, where we actually reach in and pull out their last meal. And then we also use stable isotopes in order to understand what they're eating. And what we're finding is that they actually eat lower trophic level species in general. Now, if you're out there fishing and you catch a gray snapper and you pull it past the nose of these lazy fish, they're going to take it from you. But that's not what they're typically eating. And so now I'll sort of start going through what each of the students are doing. We'll have sort of one slide each. Ori, a PhD student, is following up on some of the work that we're doing on Goliath grouper. He looked at the work we're doing with the fin rays and he says, okay, well, if the fin rays are growing in a way where you can determine their ages, maybe I can go in and look at the materials between them and look at the chemical constituents to determine what they were eating, where they were living, etc. So this is some exciting work that he's doing, and he's currently has a paper in review with that. Master's student Ben is using some similar approaches to look at another large predatory species, the Atlantic tarpon, which is a species that we know surprisingly little about, considering how economically important it is as a recreational fishing species. But he's using some lethal methods, first of all. Actually, he's using a combination of lethal and non-lethal methods. Something kills him, but so one thing that he's doing is he's using a method that was developed by Ernst and is Amy in here? And Amy Wallace. They recently published a paper on this where they can use the layers of the eye lens in order to, in a similar way as what Ori's trying to do with the fin ray, to reconstruct aspects of their life history. So that's a very exciting new method that he's applying to the tarpon. Plus he's using an extensive database of mark recapture data collected by FWC of DNA data. And those are the non-lethal ones. Butch, one of our newest members, is doing a master's degree on recompression in reef fishes. So when you pull them up from depth, if it's the wrong species or if it's too small and you have to throw it back, if you pulled it up from too deep, they might be dealing with some barotrauma issues. If you just throw it on the surface of the water, it's just going to float there and probably die. So one way that we've dealt with that is we use a venting tool, which is kind of like an ice pick, and you stab it, not repeatedly, but and then you throw it back in the water, and sometimes that works and sometimes it doesn't. And then at the very least, it certainly causes injury to the fish. And so we're looking at new ways in which to get the fish back down to depth so that they can regain their ability to swim off and avoid predators. In fact, there was a story on NPR earlier this week. Did you guys hear it? So he's using some similar methods as what they talked about in that study, or that story. A project that we just recently finished, which I'm very proud of, is one that we worked with FWRI, with commercial fishermen, and the goal of this study was to see if we can reconcile differences between fishery-dependent and fishery-independent data. Those two data sources inform stock assessment. Fishery-dependent data has really been the most important one to inform stock assessment, but we're starting to collect more and more fishery-independent data. The state of Florida is doing it. NOAA is doing it. But the two different data sources often tell different stories. And so we developed a new method to determine whether and how we can reconcile these two methods. And we're still working on the reconciliation aspect, but this novel hybrid approach that we developed is going to be incorporated by the state of Florida in their fishery independent monitoring program. And it looks like NOAA is also going to incorporate it into theirs. So this was exciting. It worked. And it looks like it's going to make a difference in how we collect our data. And it was great to work with the commercial fishermen. I think it's really important to bring them to the table. Okay, so that was the fishing branch. Now for a little on pollution. Recent graduate, Brittany, did an interesting study using the spotted sea trout, which lives in seagrass beds. It's a predatory species. And she used 3 studies, one conducted in the 1980s in Tampa Bay, one in the 1990s by this guy here, again in Tampa Bay, and then in the between 2005 and 2013. Now, during this period of 1980 through the 2000s, the Tampa Bay ecosystem was recovering, the water quality was improving, and the expanse of seagrass was increasing during that period of time. Okay, so this is another kind of good news story that we have here. And so she was looking at these 3 studies in order to see if the diet of this predator reflected these changes in the systems. And she found that in some ways it did, in some ways it didn't, but sort of some interesting results. She published one paper and has another that we'll be submitting within the next month or so. Our other recent graduate, Dinora, and current PhD student, also did some work in Tampa Bay. And this is some of her master's work. She looked at pinfish as a model species to determine what its population dynamics were like in different parts of the bay. It's kind of small here, but different realms of the bay. And then also looked to see how their mortality varied with different levels of habitat as protection embedded within these different areas within the bay. And so that gets into seascape ecology, which is an area that I think she's going to continue to pursue for her PhD. And she currently has a paper in revision. Habitat loss, I might be kind of stretching a little bit here with artificial reefs on habitat loss, but that's okay. Give me a break here. I'm trying for a theme. So postdoc Peter is currently using some really cool methods to quantify the visitation rates by boaters on artificial reefs versus natural reefs. And he's using these acoustic monitors, these acoustic receivers here, which are deployed and kept in the water. We've had them in the water for almost 2 years now, and we're finding some really interesting results. And that's that most of the boaters in this area, at least, are using artificial reefs about 10 to 50 times more than natural reefs. And so that has some important implications about the roles that artificial reefs are serving, certainly as a fisheries management tool. There's a lot of debate about the roles that artificial reefs in terms of attracting versus producing animals. But regardless of that, it's certainly attracting fishermen. Okay? If it's only producing new animals, then maybe it just takes the pressure off the natural reefs. But if it's attracting the fish and attracting the fishermen, then artificial reefs might be doing more harm than good. I'm going to be talking to the Gulf and Atlantic States Council about some of these data, and so is Peter next week. He's also using this to study bottlenose dolphins, which is why we say Fish Ecology Lab. We've known that dolphins were not fish since Aristotle. Our newest student, but certainly not newest member, Kara, she used to be our lab manager, is conducting a study to determine whether the communities associated with artificial reefs and natural reefs differ, and if they do, why. Are there different processes going on, both pre- and post-settlement? And so she's using a combination of observational and experimental approaches to test that. Moving on to the introduction side, I stayed out of this realm for a while, but then got sucked into it. If you haven't heard of the Lionfish Invasion, you've been living under a rock. The Indo-Pacific lionfish was first introduced probably off the southeast coast of Florida in the late 1980s, and then exploded in the sort of mid-aughts, like 04, 05, all across the Caribbean, all in the southeast, even up along the eastern seaboard, and now in the Gulf of Mexico. And the reason why we're so concerned is they are voracious predators on native prey. And so we're doing a large-scale study being headed up by postdoc Mark, in which we are aiming to determine at what level of effort and how often do we need to go out to keep the lionfish below some critical threshold at which they have a measurable effect on the native prey. We're